Hello, welcome to Autoimmune Voices, Threads of Hope. This is a podcast from the Autoimmune Association, and I'm Dr. Nina Luning Prak, your host for this episode. I'm a professor of pathology and lab medicine at the University of Pennsylvania, Perelman School of Medicine, where I direct a human B-cell immunobiology lab, a clinical lab, and a core facility. In my research lab, we study ways in which B cells, a kind of white blood cell that produces antibodies, can contribute to autoimmune diseases. And I also have the honor of serving as the chair of the Autoimmune Association Medical and Scientific Advisory Council. So we're raising awareness about autoimmune disease, sharing powerful stories, and building a community of support. It is my great honor to introduce our two distinguished panelists for this podcast, 2025 Nobel Laureates in Medicine, Drs. Mary Brunkow and Fred Ramsdell. So I'm going to give a quick mini bio of each of our panelists, and then we'll go into the podcast interviews. So Dr. Mary Brunkow earned a bachelor's degree in cell and molecular biology from the University of Washington, and she completed MS and PhD degrees at Princeton University under the mentorship of Shirley Tillman. After a postdoctoral fellowship with Dr. Alan Bernstein at the Lunenfeld-Tannenbaum Research Institute in Toronto, she returned to the United States, where she joined the Darwin Molecular Corporation and worked alongside mentors and collaborators, including Dr. Fred Ramsdell. Dr. Brunkow has worked for the past several years at the Institute for Systems Biology in Seattle, overseeing complex multidisciplinary projects. Our second panelist, Dr. Fred Ramsdell, earned his bachelor's degree in biochemistry and cell biology from UCSD and his PhD degree in microbiology and immunology from UCLA, working with Sidney Golub. As a postdoctoral fellowship, after completing a postdoctoral fellowship at the NIH, Dr. Ramsdell joined Immunex, where he studied T-cell activation and tolerance. Later, he joined Darwin Molecular, where he led the team that discovered FOXP3 along with Mary Brunkow. He has held scientific and leadership roles in Zymogenetics, Novo Nordisk, the Parker Institute for Cancer Immunotherapy, and he also co-founded Sonomo Bio, where he was the former CSO and is the current Scientific Advisory Board chair. So with that, I'd like to welcome our panelists into the room, and they're here. So thrilled to have you here today. And I wanted to start with just some anecdotes about receiving the news from Sweden. So Fred, let's start with you. What were you doing when you found out that you had won the Nobel Prize?
Yeah, it is the story I've told more than I've talked about the science, I think, at this point. I was off in the mountains, a little bit east of Yellowstone National Park, 8,000 feet. We got snowed on the night before, and we were in an area where there's no cell service. We've been out for 3 weeks, and my cell phone is usually on airplane mode anyway. And so when we finally got out of the mountains, we drove through Yellowstone, and my wife's phone finally picked up a signal. Mine did not. And we stopped to let the dogs run around and go pee. And my wife came out and just said, actually started screaming, and I was trying to figure out what was wrong. And she came out with a big smile on her face and said, "You just won the Nobel Prize." And I said, "No, I didn't." And she said, "Yes, you did. I have 200 text messages from our friends that said you won the Nobel Prize." And so I thought, well, apparently I won the Nobel Prize. So we still weren't really in surf range. So we drove for a couple more hours to a small town in southern Montana, checked into a hotel we were planning on anyway, and I got online and realized I had indeed won the Nobel Prize. And I started calling people. I think Mary was my second call. I called the Nobel Committee back first, but they had all gone to bed by that point. So I left a message and said, "I hear you're trying to reach me. Feel free to call back." And then I called Mary and other friends and stuff, and just started responding to text messages and emails. Then my phone began to ring in earnest. So it was a long, interesting evening.
I bet it was. Wow, what a story. And Mary, I'm guessing you might have a story like this too?
It's similar, but not the same. So I was on, West Coast time, and I'm sound asleep, and I heard my phone vibrate next to my head, next to my bed. And I just glanced at, the caller ID, and it was some, long international number from Sweden. And I thought,
Ah.
spam. And I put the phone down, and then 2 minutes later it buzzed again, and it just annoyed me. So I put the phone in do not disturb mode and really went back to sleep. And little did I know that this was happening to my husband as well. His phone was also buzzing, and he also put his into do not disturb. We're sleeping. And then a couple hours later, I became aware of kind of noises happening. I knew my husband had gotten up, and I think he knocked a lamp over and, crash. But he said, it's okay. And he left the room. And then I started hearing voices, and I thought, is he on the phone or what's going on? But then there was a woman's voice, and I thought, this is very weird. The dog was sleeping on the bed, and she just let out like one bark. So I thought, nothing too weird is happening. But all of a sudden, like the overhead light came on, and Ross came running in the room, and he said, you won the Nobel Prize. And similarly, I said, well, that's ridiculous. What are you talking about? And he said, well, come to the living room. You have to come to the living room. And there was this young woman with a bunch of cameras slung around her neck. She was a staff photographer from AP who had been sent on assignment, and it was about 3:30 in the morning. So, I mean, she was risking her life, knocking on doors in a dark neighborhood, and it took her quite a while to convince my husband to open the door for her. She had to hold up her phone she had the press release from the Nobel Foundation on her phone, and she held through the window, like your wife won the Nobel Prize. So, and she knew she wasn't really supposed to be the one to give us the news, but there was no other way she was going to get in the house. So we let her in, and then I realized, there were like 7 voicemails on my phone from Sweden. And so then for the next couple hours, it was like, wow, slowly became reality. Yeah. After having a couple phone calls and a couple Zoom calls with various people, I was like, wow. Now it's,
Oh, that must have been amazing.
Quite a morning. Yeah.
Wow. Well, thank you both for those stories. I wanted to just start our conversation today with a little framing for our listeners who are probably not mostly immunologists. So if you'll humor me for about a minute or so, let me just sketch a very brief overview of the immune system, and then we'll dive down into regulatory T cells, which of course is your thing. So let me just start with very basic immunology. There are kind of 2 main branches to the immune system. There's the innate immune system, which has very rapid responses that are kind of preformed. You have protective cells, molecules that recognize foreign invaders, and barrier defenses, and these all operate on the time scale of seconds, minutes, hours. Then you have the adaptive immune system, and that's the one that many immunologists get very excited about, that includes predominantly or prominently lymphocytes that come in 2 main types, T cells and B cells. And the T cells are thymus-derived, the B cells are bone marrow-derived. That's where the T and B in the names come from. The adaptive immune system exhibits certain cardinal features. It's inducible, specific, it exhibits memory, it distinguishes self from non-self. And because the adaptive immune system can generate a great diversity of responding lymphocytes to a particular pathogen or other foreign substance, sometimes that process results in cells that recognize self. And therefore, there need to be regulatory mechanisms that keep such rogue self-reactive cells in check and achieve and maintain immune tolerance to self. Now, as I certainly don't need to tell 2 Nobel laureates, there are several mechanisms that operate to maintain self-tolerance. And one of these is the one that the 2 of you have worked on and made famous, and that is the regulatory T cell, or Treg for short. And these cells, of course, play a critical role in preventing rogue T cells from causing disease. So I thought maybe we could turn a little bit to the discoveries themselves. And maybe, Fred, you could help us and set the stage for us and describe the origins of the field a little bit when you entered it.
So I studied originally what's called central tolerance, which is the way in the thymus self-reactive cells mostly get eliminated, but it was clear it's not good enough and you need additional mechanisms. And so we began to study those even at Immunex. How does the immune system control itself and only recognize pathogens and not your own tissues? And there were a number of different, very different processes that led us to believe there was this population of cells. And Shimon Sakaguchi, the other laureate, had described these cells in the early 90s that a lot of people basically ignored because they couldn't quite figure out how to find them in humans, and B, they weren't super well characterized, certainly not at a molecular level. But this was a very small population of cells in mice that if you got rid of them, mice got autoimmune disease. And when Mary and I both joined Darwin Molecular, one of the fundamental tenets of Darwin was to try to use genetics to explain biology in general and the immune system in particular. And the CEO of the company, a guy named David Gallus, knew about and had actually helped manage this group in Oak Ridge National Labs, which had a collection of mice that had various mutations for various reasons. And it was set up as part of the mammalian genetics unit as part of the Manhattan Project back in the 40s to understand the effect of radiation on mammals. And so he knew of this group and went there with other colleagues. And one of the mice that they zeroed in on quickly was this mouse called scurfy. And the mouse had been around since the late 40s, early 50s. And male mice, half of the male mice would die at about 3 weeks of age from rampant autoimmunity. So they had diabetes, they had psoriasis, they had, and they died. So they had all of these diseases at the same time.
Yeah.
And we knew it was one gene, it was on the X chromosome,
right.
But no one had been able to identify it because sequencing wasn't that great. And so we came along and said, well, this is fundamentally important in how the immune system is regulated. We know it's on the X chromosome. There's no gene we know of on that chromosome that could theoretically be responsible for this. So we need to figure out what that is. And so we decided this was too important not to do. And so basically we made it Mary's problem.
Well, Mary, it sounds like it's your problem to explain it to us then. How were you able to discover it?
It's my problem. So I came to Darwin Molecular with a background in genetics and genomics and molecular biology. And I had done quite a bit of mouse genetics specifically, and had a great interest in the kinds of new tools and techniques that were coming out, mostly from the Human Genome Project, which was really like gaining speed at the same time that we were doing our work. And so it seemed more and more like it was a feasible endeavor to try and identify the gene that causing this scurfy phenotype in mice. And so we brought the mice from Oak Ridge National Labs into Darwin Molecular, which was just a very small lab setup, and we converted an unused janitor's closet into a little mouse colony. At the time, you could buy these totally self-contained animal caging systems and stuff, so the air was recirculated and all that stuff. And my approach, what I wanted to do was to set up a back cross, set up a large breeding experiment that would help us really define as accurately as possible the location of the scurfy mutation. And so by generating enough animals and scoring for recombination breakpoints on the X chromosome, we started to narrow in the region of the X chromosome. And then the project became one where we had to somehow merge a genetic map, which is based on recombination distances, with a physical map, which is based on the actual DNA sequence. And so at the same time, there were other labs as part of the Human Genome Project. Other labs were doing a lot of mapping of chromosomes, and we were taking advantage of whatever information we could get from the literature and other databases at the time to generate a good genetic map across the X chromosome and put as many markers as we could there. But then at the same time, we were using resources that were coming out of academic labs primarily that would allow us to actually generate a physical map across the region as well. So based on actual DNA sequence. And by doing that, one of the really key resources that we were able to access were things called artificial chromosome libraries. So people had figured out a way to clone really large fragments of mammalian genomic DNA into a vector, either a yeast-based vector or a bacterial vector. And so by screening it, you could kind of walk across a region and generate a set of overlapping clones. And then that would give you an actual physical distance between two outside markers. And so by doing that, we ended up with a region that was about 500,000 base pairs long. And we started sequencing across that region. We also had a very good in-house sequencing core lab at Darwin. And we were, by using computational methods and cDNA screening and whatnot, we were able to get a good sense of what the gene content was in that region. So in that 500,000 base pairs, we found 20 genes, some of which were known and some of which were unknown, some of which were unknown, but clearly part of a family. You know, they had motifs in the protein structure that could sort of give you some hint as to what they might do. And then the question was, you know, which of those genes is mutated in the scurfy mouse?
Right.
And so then we needed to generate really good, high quality sequence of all the coding regions that corresponded to those genes. And so we prioritized those 20 genes based on what we thought a reasonable function would be. You know, you could prioritize the genes based on ones that would, you know, fit the bill for the kind of autoimmune effect that we were seeing in scurfy mice. And as Fred loves to say, the gene that actually is scurfy, the FOXP3 gene, was our number 20. It was the lowest priority.
Of course, the last one.
Well, all the other ones. Yeah. and that is because primarily, so this gene that we sequenced that became FOXP3 was one that had never been reported before, but it had very strong homology to DNA binding transcription factors, which from a drug development point of view doesn't make a great protein target to go after with either a small molecule or a blocking antibody type drug, because it's, you know, sequestered in the nucleus of the cell, so very hard to get to. So we didn't really want it to be a DNA binding protein, but in fact that's what it was. And it's a member of the forkhead family of DNA binding proteins, and the FOXP3 name was one that was assigned by a nomenclature committee. So when you identify new members of a known gene family, you send the sequence to a committee and they do all the phylogenetic analysis to figure out exactly where it fits into the family based on homology. And so it was named FOXP3.
Yeah. Wow. What it's funny that it's always the last one, right?
Always the last one.
My phrase usually is, I could not have been more wrong in trying to.
Yeah. So thanks, Fred. Yeah.
Well, but then over time, it's the sort of power of the discovery started to come to light, right? I mean, I guess one of the first things was maybe the linkage of FOXP3 and the scurfy mutation in mice, but then there was a corresponding human disease, right, called IPEX.
That's correct. So we collaborated with clinical geneticists at the University of Washington and Oregon Health Sciences University, who had been studying these families with children. They're young boys who get this really severe autoimmune disease that was similar in a lot of ways to the scurfy phenotype. And, you know, one of the first things we did, of course, when we cloned and sequenced the mouse version, we also identified the human ortholog. So there's an analogous gene in human. And then when we connected with these other human clinical geneticists, we were able to get DNA from them, from these the families with affected children, and we found a few different unique mutations in the FOXP3 gene in humans, and so that was very good validation that we really had the right gene, but also really kind of opened the door to helping the human disease side of things progress and start to understand what's happening in that condition.
Yeah, and it shows sort of the more generalizable nature of the finding, that it's across multiple species. And
Yes.
but then, of course, there's also the linkage of the FOXP3 as a transcription factor that is, that's linkable to this lineage of cells, these regulatory T cells. So maybe I can turn now to Fred and have you comment a little bit on the functions of Tregs and how and how the discovery of FOXP3 allowed some of those functions to be elucidated.
Happily. So one of the reasons, as Mary mentioned, FOXP3 was the last of the 20 genes, aside from the fact that it was a transcription factor that I knew nothing about and I did not want to study transcription factors, was that it was expressed at a low level in every tissue in the body, which did not make any sense to me, because this was an immunological problem. These mice died of rampant autoimmunity, so this gene should be expressed in lymph nodes and spleen and places like that. And so that was confusing to us. And there was a scientist in the lab, Rolly Kotri, who dove deep into, well, who does express this gene? And she isolated the cells that I had mentioned earlier from Shimon Sakaguchi, these rare regulatory T cells. And it was apparent quite quickly that those were the only lymphocytes that expressed this gene. And so quickly we knew that FOXP3 was unique to those regulatory T cells. Eventually we determined that it was both necessary and sufficient for the regulatory T cells, but it was a property of those minor cell populations. And it turns out those minor cells are in every tissue in the body. They are sentinels at some level, protecting immune cells from responding to your liver or your kidney or your lung or whatever tissue you want to focus on. So then it became apparent that these cells were in fact everywhere, but in very small numbers. And so that got us to understand that the reason we have these responses in every tissue is because the regulatory T cells, when the FOXP3 gene is mutated, don't exist and aren't there to prevent attacks on all of those tissues. And so this peripheral tolerance, as we call it, is completely controlled by regulatory T cells, and regulatory T cells are completely controlled by FOXP3.
That helps a lot. Thank you for that explanation. Super clear. All right. So it's a fascinating connection, FOXP3, regulatory T cells, but maybe, Mary, you can comment a little bit for patients living with autoimmune disease. What can they make of a discovery like this for, potential hope for the future?
Well, if you think of autoimmune disease as being something that's very similar to what we observed in the scurfy mice, where there's not this braking system in the periphery that keeps the effector T cells from overreacting and recognizing self proteins, then you can visualize a scenario where one way to approach treating autoimmune disease in humans would be to up the activity of these regulatory T cells. And so those approaches are definitely actively being pursued in other biotech companies as well as academic labs. And actually, Fred is quite heavily involved in those kind of approaches. Another area of disease where control of regulatory T cells is proving to be useful is in cancer as well. But in that case, it's sort of the opposite effect that you want to achieve, where in tumors, very often resident regulatory T cells are preventing the rest of the immune system from recognizing the tumor and eliminating it. So in this case, if you can remove those Treg cells, then hopefully that helps the active part of the immune system to recognize tumor cells and eliminate them. And so that is also a very active area of research.
Yeah, that's really fascinating. I think, and like you said, there's sort of two sides to every coin with an immune cell. You can, if you try to stimulate it, then it goes in one direction, and if you try to block it, it goes in the opposite direction. And Tregs, blocking them would make sense in cancer, where you want to enhance the immune response, and stimulating them would make sense in an autoimmune disease, where you want these regulatory T cells to suppress inappropriate autoreactive T cells or rogue T cells, or somehow. Well, the other thing about that I think is very interesting for our audience to know is that regulatory T cells have a sort of antigen specificity, right? So that you're not just globally immunosuppressing the entire system. You're actually going after very specific populations of cells.
Exactly.
Which, you know, is there an area within the clinical application of regulatory T cells that you think there's sort of the greatest promise at this point, or are there several in your view?
So there, as I was mentioning, there's a number of organizations, my own included, and others, that are now trying to use Tregs in various autoimmune settings. And people did this with what we call polyclonal Tregs. That is, they pulled regulatory cells out of patients, grew up large numbers of them, and put them back. And they proved to be very safe. You could see some very good biological effects. This was done in diabetes and in transplant and a number of other settings. You could certainly see biological activity and some effects, but they weren't curative, to be sure. And we also could find out that the cells would last for a year in a patient, so that was very encouraging. So what we have done, and other groups have done the same, is to put a CAR, that is, put a different receptor into the Tregs that we isolate, grow them up to large numbers, and then put them back in. So Sonoma is doing rheumatoid arthritis and a disease called hidradenitis suppurativa. Other people are doing transplant. Other people are doing type 1 diabetes. So there's a number of settings in which we understand the biology enough. There's very good data in mice to suggest that regulatory T cells can control the disease. So we're starting these trials in humans now. And a couple of the groups, have significant clinical data. By significant, I mean somewhere between 5 and 20 patients worth of data. And we're seeing some encouraging results, I would say. We're certainly seeing the cells go to the tissues they're supposed to go to. They certainly have activity within those tissues. We're seeing even some clinical responses in some patients. So, this is version 1 of this therapy. Think of whatever the phone was called before the iPhone. That might be this. We're at the flip phone stage of
BlackBerry.
BlackBerry.
Oh God, I hope it's not the BlackBerry. But I hated those things. But we're at the early stages of this. But I think it's pretty encouraging at this point. I think a lot of people are seeing things that make us believe that we're certainly on the right track, and we have a lot of approaches we know we can do to make these cells better, whether they need to live longer, whether they need to home better, whether they need to be more active. What we do right now might be perfectly sufficient, but if not, we certainly have versions 2, 3, and 4 ready to go, essentially. And by we, I just mean the community in general. We have ways to make our cells more efficacious, et cetera. So I think those are going to be the first iterations of this. What I will also say, though, what I find really exciting is that so many of the diseases that we all suffer have at least an immune inflammatory component to them. It's where the pathology is really driven by an immune response to something. So things like neurodegenerative diseases like ALS would fall into that category. Even things like heart disease and in many respects, aging, in fact, have large immune dysregulation components. And so if we can reset their response, I think there's a lot of applications beyond the autoimmune diseases that we talk about.
Yeah, that's a fantastic overview. And just to clarify for our audience, when you say that you want to put a CAR on a regulatory T cell. So CAR, I'm assuming, stands for chimeric antigen receptor, and it's the same kind of receptor that is used in oncology with CAR T cell therapy. But in this case, the CAR is to a particular, what would be the target?
So yes, so the CAR could be to a number of things. In transplant, it is usually to the organ that gets transplanted in. So whether it's kidney or liver or something. In our case, it's to a protein that's associated with inflammation. And so there are certain proteins that just come up during an inflammatory response. We've made a CAR to one of those, and we're using that. It's exactly right. The difference is in the oncology field, they put it into cells that then go kill the tumor. In our case, we're putting the same concept into a cell that's designed to turn the immune system down.
Yeah, which is a fantastic idea. And I think one of the other things in this field that's been so challenging, but it sounds like you're really getting there, is the protocols that you need to expand the cells and to keep them in somewhat of a stable subtype, so that
Absolutely.
they don't drift into other types of cells when they get into the body, because you could imagine that might be a problem.
Yeah, absolutely. And I will say that the frustrating thing about knowing you could do this 20 years ago was that we couldn't do it. The good thing is, in those intervening 20 years, we have learned a lot about these cells, about their stability, about their function, about where they go and how they respond. And so we can incorporate that as we think about how to grow these cells. We can ensure we take essentially a fingerprint of these cells, and so that we can ensure that the cells will in fact be stable and will in fact go to where they're supposed to go, et cetera. And so, we learned a lot in 20 years, and that's incredibly beneficial as we design the trials today.
Yeah. So the future looks incredibly bright, and this discovery is transformative, hence the Nobel Prize. So I wanted to end to give each of you an opportunity to comment, perhaps, just for patients, if you could speak directly to patients who are listening, who live currently with an autoimmune disease. What do you hope that they could take away from your discovery? Maybe.
Well, I guess
you can start. Yeah.
Yeah, I know. I guess what I would say is that it is still early days. I do want to remind people of that, right? These are phase 1 studies. We needed to prove that these cells are safe, and so far they look perfectly safe. But every iteration will be different, so it is very early days. But what I also want people to think about is that this is really a different type of medicine. This is not a pill you'll take every day. It is not a product you're going to inject every week or every month or every 3 months. When we dial this in, and I will say when rather than if, this should be a curative protocol. So we should have the ability to reset someone's immune system in such a way that they will not need a different drug long term. That's aspirational, but I think that's also conceivable at this point. And it may take a while to get to that point. But the point would be that we can reset this system in such a way that you'll get this drug once, or maybe once every 5 or 10 years or something. But it'll reset your immune system, and you'll go about your life without having to think about drugs at all. And you'll be walking around like the rest of us who don't have autoimmune disease.
Without the chronic immunosuppression, because it's specifically suppressing the rogue actors. Yeah, that's an amazing concept. Mary, how about you? What would you like to tell autoimmune patients?
I guess I would like to say, as a kind of non-immunologist myself, I mean, I came into the project from the genetics and molecular biology side of things, and I watch immunology, and I'm amazed at the complexity and how long it takes to really dive deep into a particular biological system that's operating within the immunology process. And it's kind of mind-blowing how complex it all is. And from the disease point of view, from the autoimmune disease point of view, I know there are, so many different conditions, and patients often have multiple autoimmune conditions going on. So everything about it is super complex. But I'm deeply gratified that like Fred said, over the last 20 years, the progress that's been made in understanding that biology is amazing and is probably exponential as, hundreds of labs around the world continue to decipher what's happening in those cells and in those processes, so that, I am confident that sometime, and it's very frustrating how long these things take, but I do see on the horizon, a time when, as Fred said, a patient can receive, a treatment that should last a lifetime, or at least, several years. And that would really be amazingly, gratifying for somebody who contributed at the very earliest stages of that, and is so important to relieving the pain and discomfort that comes along with those kind of conditions.
Yeah, that's very well said. And it is, autoimmune diseases are terribly burdensome and onerous for people who have them. And I'm sure that this discovery gives many people great hope. And so I just wanted to close before we wrap up. I just wanted to briefly thank our two panelists for their amazing contributions and for their enthusiasm. And they are role models for the rest of us to aspire to, and younger generation of scientists. Anyone out there who's listening, look at these two individuals and the amazing work that they have done. Before we wrap up, again, I just want to mention that the Autoimmune Association website has a wealth of resources. There's a lot there to help you feel informed and empowered. So please visit us at autoimmune.org and explore more. And I hope that you have enjoyed this episode of Autoimmune Voices, Threads of Hope. Please be sure to share this with somebody who might find it meaningful. And we look forward to seeing you again soon on another podcast. Thank you.